Okay guys, now we are going to talk about the classification of bacteria. We have talked about in previous terms that how we can classify bacteria in different genus and species and all these stuffs, and what are the techniques we have used. But now we are going to look at the modern day approach of classifying bacteria and how we can put those bacteria classification into a book, and in a book which is called the Bergey's Manual of Systematic Classification. In this Bergey's Manual of Systematic Classification, what we can find the information about all those bacteria which are being discovered, which are being identified as well as classified uh, throughout the time. Okay, so let me take this color for, yeah, okay, I have it. Okay, so now let's begin. Now let's go to talk about it. Now in Burgess Manual of Systematic Classification, we can find different chapters and different volumes, and those volumes are destined to put the information about a particular type of bacteria. So in this case, in the second edition of Burgess Manual of Systematic Classification, what we are having volumes, volume 5 volumes actually. In these 5 volumes, we are having different types of bacterias. Okay, they are having different types of characteristics. Now each of this volume is having a general characteristic features, and those bacteria which fulfill that general characteristic feature is put onto that volume. Okay, so in this 5 volume, the general view is that then the volume 1 is consisting of the archaeas as well as the deeply branching and phototrophic bacteria. Now archaeas are not considered as bacteria now, because archaeas are the primitive one, and as we know, if we start the journey from a common or last universal common ancestor, which we call a Luca. If we start our journey from Luca, we can have sorry, we have a branch. So you have to draw a branch from this Luca. Now one of this branch is creating bacteria, another one of this branch still start to evolve and grow, and it will grow. After some time, it will have another universal common ancestor, and from this ancestor it will branch into two parts. One of them produces archaea, and the another one produces animals or eukarya, sorry, not animals, eukarya. So that is the difference. That is how the evolution is coming throughout these three domains. So if you look at the three domains, bacteria, archaea and eukarya, that's how it's done. Nowadays, using different cutting edge technologies like DNA technology and all these technologies, we came to know that this archaea are much more closely related with with us, are much more related with eukarya than those bacteria. Okay, now in the volume 2, now in this case, in volume 1, we are dealing with this archaea as well as some microorganisms, some extremophile microorganisms like archaea. Okay, which love to be in the harsh environments. Okay, now in second volume, we have to deal with the proteobacteria. Now proteobacteria are separate types of genera because they are consisting of different types of proteobacteria, including alpha, beta, gamma and so on. So there are different types of proteobacteria we can find, delta, epsilon, all these different proteobacterias. Okay, and in the volume 3, we are going to talk about the gram positive bacteria, but which are having the low GC content. Now this GC content is really important, and nowadays scientists are very interested on this GC content, because this GC content help us to identify between different different bacteria, and this GC content also helps us to know whether two organisms are related with each other or not, whether two organisms are on the same species or not, depending upon this GC content. And we have talked this before, that if two organisms are sharing almost 70 almost sharing the 5% difference in the GC content, or less than 5% difference in the GC content, we can put them in the same species. If they are varying in over 10 to 20% of the GC content, then we can say they are on the same genus but not in the same species. Okay, so that's how we can know by looking at the GC content whether two organisms are interacting, related with each other or not. Okay, now in the volume 4, again we are talking about the gram positive bacteria, but they are having the higher GC content. That means the bacteria which denotes the very high amount of GC inside their cell, over 60 or 65% of GC inside their DNA. Okay, in the volume 5 we are talking about the planktomycetes as well as the spirochetes. And the spirochetes are different types of genera, and actually the volume 5 is made up with a kind of hodgepodge volume, because we can see that after putting all those bacteria in these four volumes, we still remain some of the bacteria which we cannot put in any of these volumes. So they are possessing some different types of features, different types and unique types of features and characteristics. So we need to put them in some volume, so they make the volume 5. In the volume 5 they put them all, so all those unusual categories in the volume 5. Okay, now let's talk about the volume 1. Okay, in the volume 1 we are generally talking about archaea. Okay, among this archaea, the one thing we are going to talk about most importantly, and which is the thermotoga, because this is my favorite one. Thermotoga is a very important one. Why? Because you can see the thermotoga structure of the cell of the thermotoga is quite unusual, because if you look at the structure, this dark black portion of the thermotoga is the cytosolic environment, and this is this part which is surrounding this dark part of the thermotoga is the cell membrane of thermotoga. But one thing is very interesting to look at this picture is that this outer layer of cell membrane is not at all attached with the cell surface, with the cytoplasm surface. So it is producing a very slight, very important and very much gap, or a huge gap in between the cell membrane and the cytosolic environment. Okay, so actually it is like someone wear a very loose cloth onto his body, so it is looking like that. So thermotoga structure is really important for that purpose. This is a gram negative bacteria. This is the rod shaped bacteria, as you can see, but this is the sheath like structure, sheath like envelope of this thermotoga, which is really important. Okay, which is really exciting in this case too. Now we can also talk about the dinoflagellates. Okay, so this phylum Dinococcus is another important part. So let me take the color. Okay, now this Dinococcus, as you can see in this picture, this is also interesting structure of the Dinococcus. We can find them in this tetrad. This in this four cells are attaching with each other to make a structure like this tetrad structure. Okay, the stain gram positive, their cell wall is layered and has an outer membrane like the gram negative bacteria. That is quite unusual. They stain gram positive, so that is fine. So if we look at them, we can call them the gram positive. But what is exciting, or some different, or different inside their cell, is that they are having the cell membrane. So normally what happens is, because of gram positive cells, we are having two layers. One is the intern cell layer, another one is the peptidoglycan layer, which is quite thick. In this case, they, although they stain the gram positive color, the stain like the gram positive bacteria, but still they are possessing a slightly outer layer, which you can only find in case of gram negative bacteria. So they are having the third layer. Okay, in spite of having this third layer, they stain gram positively. So that is a very quite remarkable feature about this Dinococcus. Okay, and we call it the Dinococcus thermus, because this Dinococcus is normally found in the thermal region, so they are having the optimum temperature slightly higher than the mesophilic bacteria, so you can find them in the huge high temperature environments. Now, if we talk about the phylum Chlorophyxi, the term which is Chlorophyxi, that means they have something to do with chlorophyll and the photosynthesis, and exactly the same, that they are photosynthetic sometimes, sometimes non-photosynthetic. In this case, we are talking about the photosynthetic green sulfur bacteria are the example of some of those phylum Chlorophyxi, and also there are phylum Chloro B. This chlorobi or chlorobi, whatever you can say, they are also the green sulfur bacteria. They are not non-sulfur, they are green sulfur bacteria. They are not capable of making food using the photosynthesis technology, but they can utilize this H2S. They use this hydrogen sulfide and sulfur as well as hydrogen as an electron source, and they can utilize them and they can accumulate sulfur outside the cell by using that. So that's why this chloro B is also important, because they are utilizing the sulfur, utilizing the inorganic matter like hydrogen sulfide to take the source as a source and utilize them to produce energy inside the cell. So that's why this phylum chloro B is also important. Now let's move on to the phylum cyanobacteria. So let's see, yeah, phylum cyanobacteria. The cyanobacteria is a very important and one of the most remarkable bacteria that we can find, because cyanobacteria is a very what you can say, it's a blaze to our civilization, because if we utilize cyanobacteria, it can give us many things. Also cyanobacteria can possess some toxic parts which which can be dangerous for us, but still cyanobacteria are the huge sources of photosynthesis, because cyanobacteria are produced enormously in the ocean environment, and there the cyanobacteria are green because they are having lots of chlorophyll inside their cells. As you can see in this picture, lot and lots of chlorophylls inside their cell. They are not only having the chlorophyll A, they are also having different chromogens like phycobilin and phycoerythrin and all different types of chromogens like blue color, red color particles inside their cell, and they are producing rapidly food. They are going through all those steps of photosynthesis and producing food and producing energy for them. Now one thing very important about the cyanobacteria is that these are the first type of bacteria which start to produce food, which start to go through this photosynthesis pathway and start to produce food. So in the evolutionary history, if you think about think about the journey from non-food, from nothing towards the production of food, towards the photosynthesis process, these are the first type of bacteria. These are the first choice of the environment who start to produce food, who start to go through the new path of producing food using the energy of sun. So that's why they are really important. And not only that, the cyanobacteria can also fix nitrogen. The cyanobacteria can also fix nitrogen, because they can take nitrogen from the environment, they can utilize this nitrogen, they can incorporate this nitrogen. First of all, they are taking this nitrogen, as you can see in this picture, and they produce the ammonia or the inorganic materials like that, and then this ammonia or ammonium compounds can be incorporated into make the amino acid, which is glutamine in this case. So they incorporate this ammonia into the glutamine, and after making this amino acid, the nearby cells can take this amino acid from these cells, so they can do this. Now the cyanobacteria can be found in different shapes and size and different orientation inside the cell. We can find them as a colony, a mixture of cyanobacteria attached with each other. We can find them separately scattered into the environment. We can also look at them, they can be circular in nature like that, they can be oval shaped like that, they can also be fibrillar structure like that. Okay, so whatever we are looking at, they are having we can also having the cyanobacteria which are having the specialized cells. We can also having the cyanobacteria which are not having the specialized cell. So most of the time when the cyanobacteria have the specialized cell, they can produce two different types of specialized cells. One is called actually I forget the name of that, but one type of cell is so much durable in the harsh environment or the environmental stress. They act like a spores of for the bacteria. They can stay for a long time on the environment. When the environmental situations turn to their favor, they can germinate and produce new cells. And you can also find another specialized cells that you can see here, which is called the heterocyst. Now heterocyst is a specialized compartment inside the cyanobacteria which start to uptake this nitrogen, convert them into ammonia, and incorporate them into this into this amino acid, and thus by utilizing the energy from the nearby cells, which are the normal cells or the vegetative cell, this heterocyst cell can utilize them and incorporate the nitrogen into the cell, and give this nitrogenous compounds, give this amino acid to the nearby cells, and in turn it can take the energy and carbon dioxide from the outside cells. So these are the arrangement of work, and very superior quality work we can see inside these cells of cyanobacteria. The arrangement of different types of cell and specialized types of cell to do different types of works, to do different specialized works, that is a really very important thing and very updated thing, okay, I must say. Okay, if you know you want to know about the phylum cyanobacteria, you can turn on to the cyanobacteria turn on to my discussion about cyanobacteria. I have a separate video about